us, we should be. And you can ask them questions too. Okay. You should. I'll do my best. Grill them. No, I'm just kidding. Okay, are we ready?
I think we're ready. Are we ready? Good morning. Come on, show everybody what we've been working on all week. Good morning. All right, there we go. Good morning, everybody. Students, welcome to the last day. Parents and everyone, I guess we have a handful of folks watching us online. Welcome to the summer wrap-up of Nanoscale Microscopy here at the California Nanosystems Institute. My name is Adam Stieg. I'm Associate Director of CNSI and one of the instructors of this program. This is Rita Blake, my colleague, who is our Education Director. She'll sort of give you a sense of all the folks that have been involved in the program this week. So I'm just here to say welcome to UCLA, welcome to CNSI. It's been fantastic for the students to have you all here. We did this program for a number of years. We put it on hiatus, I guess, for what, the last 2 years, as many things have gone on hiatus. We really couldn't do this class remotely because of the hands-on component that you have to really learn not just about how microscopes work, but sort of see them in action. So it's been fantastic to have you all back here. I know it's been a week. It's like a whirlwind. I woke up this morning thinking, how is it Friday already, right? Like we're done. But nevertheless, I hope that you had a good time. You had an opportunity to meet some new friends and learn some things that hopefully you can carry on when you go into the rest of your educational endeavors. For all the parents, thank you for sending these bright young minds here to share the week with us. It's been exciting. Lots of fun interactions, good questions, and hopefully some really exciting presentations that we're going to see, sort of culminating and synthesizing all the things that they learned about. Well, maybe not all the things that they learned about. I think we sort of had a bit of a fire hose going on of content and information. But I hope that we'll see some nice distillation of some of the things that we've exposed the students to this week. And putting them up here in front of everyone to give a presentation is always a bit nerve-wracking for them, but I'm sure you'll do great. I did see some of the slides. They look quite good. So I will turn it over now, just saying again, thank you and welcome to the program, over to Rita, who will introduce sort of, the background of the folks who makes this happen and what the students have been up to this week before we turn it over to the real people you want to hear from, which are the students. So.
Great. Thanks, Adam. So, like Adam said, my name is Rita Blake. I'm the Director of Education for the CNSI. So I have the pleasure of seeing all of the summer programs and helping out with all of them. And like Adam said, I really love this program as well because it's got such a unique hands-on component to it where these students have been incredibly lucky over this week. I mean, they got to look at and actually work with instrumentation that is so advanced that even a lot of graduate students don't always get to work with these kinds of tools. And so I think it's such a great opportunity, so I'm really glad that we got to do it again this year. And there's quite a lot of people that were involved in making this happen, so I'm just going to start out by introducing some of them. So you all already met Dr. Stieg, but they've had several instructors over the course of the week. So one of whom is Dr. Laurent Bentolila, who is right over here. He's the scientific director of the Advanced Light Microscopy Lab and a researcher here at the Institute. So he introduced all of the students to different techniques involving advanced forms of light microscopy. So things that you will be hearing a lot about, so I will not introduce them because I'm going to let the students introduce them during the presentations. They also got an introduction to electron microscopy techniques from Dr. Matt Mecklenburg, who unfortunately cannot be here today, but I know sends his regards and hopefully is still getting to watch the presentations from the live stream. And we also had throughout the week Brian Zhang, who was our teaching assistant, helping all the students throughout the week, helping with taking them through the labs, reading their blogs, answering questions, pushing their projects to be the best that they could be. So you'll probably be seeing Brian during these presentations as well. And then there were a lot of people working behind the scenes to make this program happen this week, and I'm going to introduce and thank all the rest of them later. But just to reiterate, again, I'm Dr. Rita Blake, and then also our program coordinator, Dr. Elaine Morita, has been critical in just all the behind the scenes stuff of getting everything and everybody here to make this program happen. So I want to give an extra thank you to Elaine. So thank you. So just to give you all a little bit of context before we go into these presentations, we have two goals with this course. So one is to give students real hands-on exposure to advanced microscopy, because it's definitely something you can read about and know theoretically, but it's very, very different to get to experience them firsthand and really see how they work. But then all of these techniques are fantastic, but they don't operate in a vacuum. I mean, the electron microscope technically operates in a vacuum, which you'll learn about, but they don't operate for no reason. So what we really want to do is get these students to think about how these different microscopy techniques can be used to actually solve real world problems that scientists and engineers are thinking about. So that's what you're going to be seeing during these presentations. How these advanced techniques are capable of solving problems and where maybe new techniques might need to still be developed. And there was a fully packed schedule for this whole week that you can see a snapshot of here, where the students had 4 full days of just so much exposure to techniques and some amount of time to synthesize them. And I know that they've been working very, very hard, including last night, to get these presentations together. So I am particularly excited to see what you all have come up with. And we're going to be asking you some tough questions. And we're asking you the tough questions as a learning experience and an opportunity to really, see what you've learned and help you guys see what you've learned in this very short amount of time that we've had together. So with that, I'm going to stop talking and I'm going to turn it over to our teams and have them start talking. So team 1, can you all come up, and we're going to start with you. And let's give all our teams a hand.
All right, hello everyone. We're team 1 and we're going to be presenting about infectious diseases, and specifically on how electron microscopy can help to develop better techniques and to make it more effective. So first of all, let's introduce ourselves. I'm Mark.
I'm Jack.
I'm Greg.
I'm Grant.
Oh yeah, and here's a table of contents.
Okay, so most known biological materials, such as viruses, can take less than 10 beams of electrons before fracturing. So to combat this issue, scientists today prepare tens to hundreds of thousands of viruses just for one complete image, as one virus can only offer about a pixel of view. After scanning, the average of all the scans is taken to achieve atomic resolution. The current method may take up to weeks for a complete image. It may also lead to inaccurate results, as not every virus sample is identical. Although close approximations of virus structures can be achieved with the current technology, exact imaging is still unachievable. Therefore, new technologies that do not damage the sample when scanning would be needed. This would mean that the EM only needs a few samples, saving both time and accuracy.
Right, so you can see the electron microscope on the right. Electron microscopy is a technique of microscopy that is similar to an optical microscope, except it uses electrons instead of light. And it is extremely important when imaging biological materials, since it's the only way to really see in-depth 3D nanoscale biological samples. And an important focus point of infectious disease is the interaction between viruses and the host cell. It's called glycoproteins, and it's important because it's where the antibodies bind to the virus. And these interactions occur on an atomic level, so it's important to use an EM to image them.
Okay, so electron microscopes play a major role in the creation of vaccines and antibodies due to their ability to create high-quality images of viruses. something which other microscopes like optical are unable to achieve at such a level. So these higher quality images have been crucial in discovering how each virus works, and how researchers could possibly combat their method of attacking host cells. So the way that the EM works is by shooting out multiple electrons at a specimen, which is prepared in such a way that allows the electrons to pass through it. So these electrons shot out are condensed and pass through the object, which is then magnified by multiple lenses before coming into contact with a fluorescent screen. This screen will glow brighter the more energy the electron hitting it has, which will result in an image showing up on it. And that's like a diagram of the thing, the electron microscope.
So as stated earlier in the abstract, my bad. So
Just leave it.
as stated earlier in the abstract, the current issue with EM is that biological samples are easily destroyed by the electron radiation. However, in contemporary technology, EM does offer some workarounds to lessen the degradation of biological samples, such as cryo-EM, lowering the electron intensity during observation, and using multiple samples. However, each compromises does come with its own disadvantages. For instance, cryo-EM freezes the sample when taking its models. While this does prevent the samples from taking secondary radiation damage, it sacrifices the biological accuracy of its models, since samples will look different when it's frozen compared to its normal state. Lowering the electron intensity during observation does help decrease the degradation rate of biological samples, but it comes at the expense of lowering the resolution of its models, which would increase the time it takes to make a high-resolution model from hours to a few days, and can even be up to weeks in the higher spectrums. Using multiple images to render out one model is also a workaround to lessen degradation damage. But however, this just like cryo-EM, this also sacrifices accuracy, since averaging thousands of shots of different virus samples negates the fact that there are slight phenotypic differences in each viruses in each sample. So the top image here shows undamaged herpesviruses, which are typically the ones that are going to be used during 3D imaging and making high resolution models. And the bottom, you can see that these are heavily damaged herpesviruses, and the membrane of these viruses are basically completely destroyed from electron radiation, which is the main problem that we're facing, and it makes it unusable for any 3D modeling for research purposes.
So you may notice that most of the workarounds to damage of electron radiation does not actually deal with the speed of electrons or how the electrons damage the sample. And really the reason that electrons damage the sample is because of the speed of a particle that has mass. So there actually is a type of microscope that can help with the effect of electrons on the sample. It's called an environmental SEM, and basically what it does is, instead of putting the sample into a vacuum, it puts it into a chamber of water vapor. And the water vapor can slow down the electrons before it hits the sample. And scientists have found that the ESEM produces less E-beam-induced artifacts, so basically it damages the sample less than a traditional SEM microscope. However, there are drawbacks. So if you look at the images on the top, these are 3 images of the same thing from ESEM versus these 6 from a traditional CEM microscope. And there's much higher resolution in a traditional CEM microscope compared to an SEM. So you have about 5 nanometers of resolution for the ESEM compared to 0.2 nanometers from TEM. So that's really the resolution that you need to see the interaction between the virus and the host cell. However, we can get some information from this, so we know that slowing down the electrons isn't actually a viable way to reduce the damage to the sample.
So like just to combat this issue, like how like ESEM have like lower resolution and everything. So the new solution that we came up with is to implement a magnetic field in the middle of the EM, which is like around right here, which is like red boxed, to slow down the electrons. And this does not need any water vapor, so like the resolution is not going to be as damaged. But also, just to implement the field like this would be also like much elongated, like to the design of the full microscope. So like if this concept like is successfully implemented, the degradation rates of biological samples will be like not as fast as before, and it will be more accurate since like one sample, one virus can like be taken more like pictures and like with more electrons. And like this also offers like also a big advantage is that the ability to adjust like what you want. So like for like every single biological sample is different, and like you can adjust the speed of electrons just to sacrifice how much resolution you want or how much damage you want to be done to the sample. And so here's the future directions. So it is a great concern that like right now, as said before, thousands of samples are used to take for one image, which like we may introduce like inaccurate results. And with our new technology, we can have like less inaccuracies because it only uses fewer samples and in much faster time. So like some impacts of this may lead to like faster vaccine development because like that's going to take much less to understand the virus fully, and then like less expensive image because like right now like the rates to use electron microscope can be like up to a few $100s an hour, and then it's like the queue time, like many people are lined up just to use them for like their own research, so like that should be some impacts. And right now on the image is like what the typical virus TEM scan looks like, which are after like thousands of averagings. And this is our references. And thank you. So does anyone have any questions?
All right, we want questions, particularly from students, if we can. Parents are great. Students. Rita has one.
But I'll let other people ask first.
Do any of the students have a question? This has been my challenge all week, is getting them to. All right, well, if not, Rita.
So I have a clarifying question. So in your abstract, I think you said something like viruses can only take up to 10 beams of electrons. Can you elaborate more on what you mean by that? Because that phrase feels a little vague to me.
I think it's from the lecture given by Dr. Matt. I think he's talking about 10 beams of electrons per square angstrom. Or I'm not particularly sure of the units, but like after a certain intensity and concentration of electrons shot to the virus will explode just like the image like Jack talked about. Yeah, like this image. So this image was actually like only 2 to 3 seconds after like this. It's only 2 to 3 seconds. So like after like long intensities, they will just explode.
I have a question. So first of all, a great presentation. Thank you. And I wanted to ask you, do you have any idea of the magnitude of the electromagnetic field you'll be applying to slow down the electrons?
I think the problem that we saw with the environmental SEM is that you can't really adjust how much the water vapor slows down the electrons. So I think we were thinking that the electromagnetic field would be adjustable, so we can decide how much resolution we would sacrifice for the less damage to the electrons.
And it can be adjusted like on a case-by-case situation.
Of course, I think that the actual magnitude would be pretty slight, since you wouldn't want to be slowing it down like too much, since like the fluorescent screen would be pretty dim if it's like super slow.
Any other questions?
Parents are good too. There
Okay.
we go.
This is more of a mom question, but were you exposed to these specimens, like the herpes? Or how did you actually get these images?
So we were actually, Bruce showed us these during our rotation. So we were, I took these 2 pictures on my phone when the samples are in the microscope. So I think it's completely safe. So, yeah, but yeah, to answer your question, we were exposed to these. I mean, not exposed, like we did see these images, these viruses in person, yes.
Although I would have liked to be a little bit closer to them. It would have been nice to like get a better look at how the samples look. But yeah, we should be safe. I'm hope.
So
We might
it
take
was
a test later.
safe, guys. Okay. Just for your clarification, they were definitely interacting with real world samples, right? And in many cases, those samples, if they were out in the environment, would potentially be hazardous, right? But all the hazardous things were handled by our professional staff. These samples were inside of the microscope, already prepared, and basically they're no longer in an infectious state in this case. But there were definitely real world samples. There were live cells in some cases, things that they were working with. But the curriculum is designed to put them in a place where they're seeing important, like interesting things, but to do it safely. Okay. Well, if there aren't any other questions, let's thank Group 1 again. And we will transition to Group 2. So why don't you guys close down and Group 2 head on up.
Oh, you gotta mute it. My bad.
How do I? Wait. It won't. Oh, I have to look at it from here. Click out of it. Can I just like close it?
And then double click on yours.
It's not. It doesn't show up on.
Because you just have to move your mouse, the cursor over. Do you want me to help?
Yes, please.
Yeah.
Well, because it's on the live stream, we need the audio for YouTube. So if you want to do it, there's one here, and you can also use this one.
Yeah. Okay.
All right. Okay. Good morning, guys. We are Team 2, and we are here to talk about nanorod herpes treatment.
Okay. So we decided to choose beta herpes virus, and it is part of the herpes family. And during the latent stage, it can go back to reactivation. Like reactivation can be provoked by external and internal signals. And we use cryo-electron microscopy to create a drug that can be a treatment and can undergo endocytosis to target infected cells. And an improvement that we can have for cryo-electron microscopy is to manipulate thickness of ice. And the drug contains a coupling of the nanorods and antibodies to target specific proteins that this particular virus creates. And gold nanorods do use optical lighting, and the main goal for this project is to find a therapeutic drug.
So for our area of focus, we wanted to focus on electron microscopy. And electron microscopy basically works by an electron gun, as you can see in the diagram. It shoots electron beams down into a sample, and that way we're able to get images. However, because the electron beams can sometimes be really intense, that can cause fragile materials such as biological matter and biological materials, which what we're working with, to fracture and even corrode. So the solution to that is using cryogenic electron microscopy, also known as cryo-EM. That entails basically dipping the sample in virtuous ice, and in that way we're able to protect the biological matter. And if it does fracture, it's holds still in order to reduce noise and distortion within the imaging. So we're able to get high resolution imaging, such as like over here where it's a close-up of a capsid vertex, which was achieved through cryo-EM. And then for our other area of focus, we wanted to focus on infectious diseases. Earlier within the session, we learned about bacteriophages and how that's a natural way of combating bacteria, and also about the artificial pyacins that are being engineered, which will essentially depolarize bacteria in a non-invasive way. So in order to render it useless, basically. we wanted to take those principles but repurpose it and take it a step further to target viruses rather than just bacteria. So the specific virus that we wanted to focus on was herpes virus, and this is because herpes virus is extremely challenging to try to handle. It's constantly evolving, and as you can see, its large size allows it to have a variety of genes, which allows it to have hundreds or thousands of proteins being produced, and this high protein production makes it very challenging, especially compared to influenza, for example, which only produces like dozens of proteins at a time. And so for the stages of herpes virus, not only is it hard because of all the proteins, but it also is a latent virus. So that means that when it goes into the immune system, it is able to produce all those different proteins in order to evade being detected by the immune system. Afterwards, it would go into the lytic phase where it essentially causes reproduction within the virally infected cells. Once the immune system recognizes that that is happening, it produces antibodies, but before those antibodies can become effective, the herpes virus would basically go into the latent phase in which genomes become tightly packed and proteins are no longer produced, rendering the cell or the virus itself undetectable. And then from there, it can go into animation and reactivation later on in life.
Okay, so as of right now, there are no cures or treatments for the herpes virus, but there is ongoing research talking about vaccines and cures. Actually, currently right now in UCLA, there's herpes research going on. You guys probably know about this through the VR lab program that we did. And so they were basically investigating KSHV, which is a type of gamma herpes virus. And the most important part of it is a portal vertex complex, which basically pushes DNA and packs it very tightly into the virus and basically nucleates the DNA. This is made up of 5 subunits, and it's actually asymmetrical relaxed structure, and it will insert 150 kilobases of genome. So over there's over there's like a picture of a very like tightly packed virus. So basically what is happening right now is that they are in the lab trying to manipulate and cleave the vertex portal from the virus. And they basically clip the beta sheets, and this will actually separate the 2 bodies apart from each other. And this causes the virus to basically terminate because it no longer has the DNA. The second stage is they genetically engineer model proteins that take the shape of a vertex portal, and instead you will end up having an empty caspid without DNA in it. Okay, so I'm just gonna briefly go over this, but basically, as you said, there's a lot of problems when it comes to cryo-EM, and one of them is you can't get a very clear image because of the thick crystallization of ice. And go on. So basically, this is just a picture of showing like what cryo-EM is, and a lot of the times, like most common, it scans between 200 kilodaltons or larger, but herpes proteins are actually 92 kilodaltons, so it makes it a lot harder to get high resolution. So our solution is to use nanotechnology and a nanowire. Basically, this is a hydrophilic wire which will help cut the ice into thin slices, so we can reach the border between having enough thickness to protect it, but also enough thinness that electrons will be able to transmit and produce a clear image. Okay, so we took inspiration from cancer chemotherapy. And so how have scientists used targeted therapy to kill cancerous cells? So they use gold nanorods, which basically they gather our localized spots in the cell, and because it's a conductor, it will actually be induced with heat generated near infrared light, which basically what happens is the light energy will oscillate and then turn into heat. So the heat will actually kill any structures that are near it. So scientists start to target proteins that will help proliferate cancer, help move it faster. So if we are able to stop these proteins, the cancer will actually die. We are repurposing this method, but focusing on the herpes virus.
So our solution
with antibodies that can enter the bloodstream, and these antibodies can target virally infected herpes cells. The antibodies will be carrying gold nanorods, as previously mentioned, and they will go and target and attach to infected cell receptors. When superheated, the gold rod will destroy the virus, and antibodies, modified or natural, can detect the infected cells. This theory works by first detecting the virus in the latent active state, which circles back from the, the virus from the latent active state and then circles back to the latent stage. Antibodies will carry the nanotubes, and when released at the cell membrane, and then will be released at the cell membrane, and this is the start of the process of endocytosis, which is that image that you see on the right side. The rods will cross through the cell membrane and bind to herpes proteins, and the function of those proteins is to evade natural antibodies from detection, and we do not want that. The way to activate these rods is with PBT, or photothermal therapy, which is pretty much an optical light concentration at the nanorod, and this will allow herpes cells to be detected by the natural body system. And these nanorods are a better way to treat herpes virus because of its unique structure that will be able to produce effective results while retaining the nanoscale perception. Nanorods are also less invasive, meaning it won't easily spread to other proteins that we do not want to be destroyed. And these gold nanorods can hold large amounts of heat, making the rods extremely hot, and when heated with thermal therapy, it uses near-infrared light and activates the gold rod. And these rods have very specific targets, so it only locks onto a certain protein and it sticks. Okay, so future directions. So these are the various types of viruses or herpes that are here, and basically we want to take our solution, which is our drug, and basically treat all these herpes because let's take an example like cytomegalovirus. Basically, beta herpes virus, we target immune cells, and for the cytomegalovirus, we target epithelial cells. So it's basically, we use the same type of treatment, except instead of targeting immune cells, we target the cells that that virus it uses. So it would be easier to just use that treatment. Okay, so for our other solution, we were thinking of iron oxide particles and how we can put that in drugs. And this could go into the neuron because herpes hide in the neurons. And so we could use MRI waves to kill this or to activate the iron oxide and essentially damage the DNA. However, we can't do this because if we do that, then the neurons will also get damaged, and that we don't want to do that. So basically, it would be revolutionary to essentially just find a way to emit or to activate iron oxide without damaging the neuron.
And we wanted to say thank you to the staff here at the California Nanosystem Institute. We also wanted to say thank you to Michael Lake and Jonathan Jiff for helping us gain a really deeper understanding into our idea and our concepts.
Thank you. So I was really interested in your ice cleaving technique with the nanowire or nanorod or whatever you were using, but I was having a hard time sort of understanding how you would actually use it to cleave. Like, can you sort of describe the process of actually cleaving the vitreous ice with this technique?
Okay, so it's a kind of new subject, so not a lot of people know about it, but it has I think it has weighted ends, and when pressure is applied, it can cut exactly how much you want it to cut through. And because it's hydrophilic, it's able to slice through more easily, but vitreous ice is different from regular H2O ice, so it may not work
But like, is it like attached to something that is larger and you're like physically trying to do it? Are you using it like in a machine that's like digitally controlling it? Like how is this actually literally applied onto your ice?
I was thinking like a machine where like kind of like microscopic where you get to manipulate atoms, but this time we're manipulating the bar where it can like move across.
Do you think that a scanning probe microscope could help with that process?
It's an engineering problem. I mean, I've never seen this before. It's interesting.
Yeah, it is interesting.
I also had a question about this slide. Like you have on the slide, there was like previously the manual blotting of excess fluid. Can you just like describe that? Is that just not as accurate? Is that why you're trying this new solution?
I think they were like trying to create methods of like just freezing it, but then they had to manually like even it out and get rid of the excess fluid. So by using like a machine where you can calculate how much you want the thickness and the nanometer, we can get a more accurate and a more clear image.
Thanks.
Okay, so the start of the talk was about solutions to enhance the performance of cryo-electron microscopy, right? And then you went into gold nanorod, antibody-bound nanorods for therapeutics. Both interesting topics. I'm trying to understand the connection between the two, right? How does cryo-EM help you with the nanorod research, right? What is the enabling feature of the electron microscope that you would use to help you develop these targeted nanorod therapeutics?
It's like we use cryo-EM to understand the 3D structure of what we're working with. So we were thinking of because we have to genetically engineer the antibodies that carry the nanotubes, by being able to understand like what we're targeting and how we're gonna manipulate our genetically engineered system to target the herpes infected cell. Like that way we could use cryo-EM to be able to do this, get a more clear image of what we're doing.
Okay. All right, we got a question back there. Student question. All right.
All right. So I had a question about you were talking about the iron oxide, right? Which is activated with the electromagnetic waves. Like, how does the mechanism for the iron oxide active? Well, first of all, what does iron oxide activation mean in the first place? And second of all, what's the mechanism for its destruction of DNA?
Okay, so basically iron oxide will heat up, and this will cause it to basically activate, which will like damage the DNA like around it, or like the localized area.
It's essentially the same thing as the gold tip nanorods, except rather than using the gold nanorods, we use iron oxide because it's less invasive within the neurons. And it's still in early development also.
And you can get deeper in the brain.
Yeah.
All right. Thank you.
One more question, and then I think we will move to the next group, but there is one more in the back corner over there. So.
And then also to the presenters, the AV team just said we have to speak into the mics. So if you're answering questions, please use the mics. The live stream can't hear you. They're getting comments.
Hello. So you guys mentioned that herpes viruses have like a huge genome. There's a lot of proteins that are produced by or coded for by the herpes virus genome. So if you guys had to target like a specific protein or a specific set of proteins with your antibody development, what types of proteins would you guys choose?
That's a really specific question. So because there are like hundreds of thousands of proteins being produced by the herpes virus, as you stated, like in your own research, like the protein structures that we're able to map out through cryo-EM, from there we would be able to specifically target the proteins that we hope to target the proteins that essentially enable them to get past the immune system undetected in those early stages of development. Like, if we go back to the so in the first couple stages, in the first step, that's when the herpes virus is still in its early stages of infection. However, if we target it through there, it's a lot harder to detect because it's not as symptomatic. So instead, we hope to target it while it is symptomatic and while it has already infected a lot of the cells. And that way, a lot of the proteins are being produced relate to the cells. So with the beta strain of herpes virus, it produces skin lesions that we can essentially minimally, like, invasively introduce the golden nanorods through that way. So this is specific to the beta and the gamma strains of herpes virus, in which we essentially just wait for it to be symptomatic, and that is when it's at its most vulnerable state, and it's less likely for it to go back into the latent phase at that time.
All right, thank you.
Okay.
All right, team 3. All right.
Hi everyone, our project is on the Brain Initiative. I'm Megan Werner.
I'm Eva Matencian.
I'm Emily Bautista.
And I'm Francisco Cardenas.
So the Brain Initiative is focused on providing a clear map of the brain on the nanoscale. As seen in the picture, 1 neuron out of 4 billion is connected to 4,000 axons. This in-depth structure of the brain has made it difficult for microscopes to proficiently capture each neuron and connection. Because of the limitations of microscopes to map the brain, mental illnesses and disorders such as schizophrenia, Parkinson's disease, and epilepsy remain uncured. Designing an efficient microscope to map the brain by each individual neuron and connection will lead researchers to understand the brain. By understanding the brain, major advancements toward drug cures are achievable.
In order to see the brain at a more in-depth scale, light microscopes are most commonly used. They work by bouncing a laser off a sample, then focusing the light through an object and some more lenses in order to magnify an image. Light microscopes are limited thanks to the limit of the visible light wavelength spectrum. There are many techniques which have facilitated the usage of these. For example, fluorescence is a way in which researchers are able to highlight specific parts of a sample in order to make them visible under a light microscope. This can be achieved through either changing the genetic information so that neurons or other biological samples such as cells produce a protein that can glow a certain color when light is shown on it. Researchers also have non-harmful synthetic materials that can do this. Confocal microscopy is a way to focus light from a sample into your eye better. It utilizes a pinhole in order to get rid of out-of-focus light. It essentially just presents better images. Like, for example, this image is of neurons taken by a confocal microscope.
Okay, so just based on the goals of what the Brain Initiative and like what we currently understand about microscopy, we created the question, how can brain diseases be cured or prevented in a non-invasive way? And the 3 brain diseases that we wanted to focus on specifically were Parkinson's, schizophrenia, and epilepsy. And we were going to focus on how our understanding of like optical microscopy has helped us understand these diseases a little bit better. So the first one is Parkinson's disease. So what it is, it's essentially like a loss of functional nerve cells in the brain responsible for producing dopamine. So in the image to the right, there's this little like black subsection of your brain that's called the substantia nigra, and it's basically the location where nerve cells that deteriorate from the Parkinson's disease, that's where they're located. So currently microscopy is being used to understand this disease a little bit better in hopes of finding a cure. So what some researchers have noticed is that patients with Parkinson's disease sometimes have affected eyesight. They have something called like convergence insufficiency, which is essentially where you like get double vision, so you see two of everything. And so researchers wanted to explore how we could use corneal confocal microscopy to track the progression of Parkinson's disease within patients. So they wanted to try to use it as like a biomarker for this disease. So after they did the experiment, what they noticed is that corneal like nerve fiber length density in area and like branch density was lower in patients with Parkinson's disease versus like those without it. So essentially what all of this is saying is that we're currently using confocal microscopy to like understand this disease better, but there's still no cure. So why is this? Well, right now we have treatments to like try to offset some of the symptoms of Parkinson's disease, but there's no way to actually like stop the progression completely. So you can do a medication to like increase the amount of dopamine, or you can do like deep brain stimulation, which is basically like a brain surgery where they implant electrodes into your brain. But like back to why we don't actually have a cure, it's because what we do know is that patients with Parkinson's disease have neurons that are affected by a certain protein called alpha-synuclein, which like alters cellular pathways and aggregates in host cells. But we just don't know how to stop this specific protein from affecting neurons because we can't tell which ones are defected. Just because like our technology is limited right now, that's just not something we're able to do. And so Eva will talk more about this later, but like one of our solutions that we want to try to do is to create an artificial neurotransmitter that like we can control like a receptor to like monitor and control the activities of neurons and cells. Because hopefully if we're able to do that, we're able to see like which ones are defected so we can like create a cure for Parkinson's disease.
Okay, so similar to Parkinson's disease, schizophrenia has no cure, but there are alternatives to suppress the symptoms. Schizophrenia is thought to be caused by a change in the levels of the neurotransmitters dopamine and serotonin. From mutations, the structure of DNA is changed, and that implements a new function. And so the resulting function has proven inadequate to the conventional structure and function, as the nerve cells have been unable to produce enough neurotransmitters. Brand scans from a positron emission tomography have shown that those with schizophrenia have lower levels of proteins found in the connections between neurons as a result of the mutation. As you can see in this image over here, those with schizophrenia have reduced brain connections. So drugs have been produced aimed to improve dopamine levels, but as they weren't completely effective, other drugs are being tested that target other neurotransmitters. And it is a complex and varied illness, and researchers don't know enough about the brain to contribute a solid cure yet.
Epilepsy has been found to be caused by the chemical imbalances of calcium ions within neurons. Scientists try to prevent epilepsy mainly by stopping seizures from occurring in the first place and from preventing their spread. Preventing can be achieved through drugs that deliver calcium ions to VDCCs, which are voltage-dependent calcium channels. However, these drugs are not perfect, and often seizures can still occur. Preventing the spread of seizures is possible through optogenetics, which is controlling neurons using light. However, actually implementing this long-term can be challenging. It requires like a big rod to stick out of the head of, in this case, mice. And neurologists do not actually know which neurons control what yet, so it's difficult to actually use. Also, it's mostly only been tested on mice, and going from mice brains to human brains is too big of a step. However, if advanced enough, optogenetics could stop seizures in general from spreading. It could be really useful. However, just research hasn't been done yet. And this image shows how fluorescence was used to determine which neurons activated during the seizure.
Okay, so to this day, understanding the brain remains an elusive, distant frontier, and it's difficult to cure the brain-based pathologies previously mentioned without understanding brain circuits. Many of the technologies we have at the moment have a limited view.
Measurements from individual neurons do not give insight into our processing because circuits operate at an emergent level, which means that brain functions rely on interactions from a large population of neurons. Now, this doesn't mean that magnified images of neurons aren't important, but rather microscopes need to have the capability to monitor different areas of the brain in parallel. Another issue with using confocal microscopy is recording data deep within the brain since they need light to operate. And as you know, the brain doesn't have built-in LED lights, but an optical microscope is the safest way to magnify activity within the brain. Electron microscopes would be great because they don't require light. However, the use of electron beams is too damaging to use on live samples. And okay, so what if we had a device that could take data from inside the brain rather than scanning from the outside and has the ability to track the electronic pulses of large population of neurons? So this exists in planar electrode arrays. However, these devices suffer longevity issues, and because they are invasive, it causes inflammation in the brain and it interferes with data collection. Furthermore, currently a system that marks different neurotransmitters fails to exist. Developing images of the 10 nanometer synapses is great. However, we can't further our understanding if we don't have a way to identify the various neurotransmitters. Basically, it's very easy to get lost in the brain without a way to decipher between the different signals being passed through. So what can we do to solve these limitations? Alrighty, so we need non-invasive technology that can simultaneously collect data from individual and whole networks of neurons. And from this data, we need to establish identifiers for specific neurotransmitters. Perhaps we can establish this utilizing two-photon microscopy, which uses longer wavelength photons to penetrate more deeply, and optogenetics, which is a method for controlling a neuron's activity using light and genetic engineering. Optogenetics is also promising because it can enable phototagging, which is a method of identifying genetic phenotypes of neurons, which is great for adding classification while recording the wavelengths occurring in the brain. Also, we believe that developing artificial neurotransmitter receptors that can monitor and control activities of neurons could provide the best insight into the brain and could be used in feedback circuits to control diseases such as Parkinson's or schizophrenia. All right, here are our resources, and thank you for listening. Any questions?
Yeah.
We could hear too.
Here, hand it here. In the middle there. I saw it there.
I was wondering, like, what types of samples would you need to measure, like live brains or like cut samples or something?
Yeah, so that was actually one of like the big limitations of the Brain Initiative is you need live samples. For many of these diseases, it's the you need to see the neurons actually interacting with each other to understand it. So for many ethical reasons, it's hard to take those measurements in live patients.
Yeah, so like a live brain sample.
So actually, I would like to follow up on this question. From what you have seen during the week, especially when you went to the different labs, what can you think about to answer that question? Let's forget about the patients. How can we kind of learn things without necessarily working with patients? What type of other models can you think of?
Interestingly, I actually read an article where they made a handheld confocal microscope that they were using for neurosurgery. So they brought it to because I know looking at the lab, seeing these big microscopes and how technical they are, we can't just fit someone's head under the microscope. It just wouldn't work. So it was very interesting to see that this introduces like a whole new realm of microscopes. Is instead of developing it, or it's like developing it around the sample, so making it fit the sample. So that's like a new realm of research.
Okay, great. But what about the little fish we saw in the lab?
Oh yeah.
Right?
Yeah.
What can you tell us about the little fish?
Okay, so using like other animals to study neurons and then relating it to the human brain.
That is right.
Yes.
I think for the sake of time, we're going to move to the next group. Well done. Thank you.
Thank you.
So as Team 4 comes up, I just want to ask, a quick reminder that the presentations are supposed to be at 8 minutes, and I think a few groups are going over. We love the detail. It's super fascinating, but just try to be succinct, because we want to be sure to get all of you in and be done on time.
Because no one wants cold food.
Okay. Oops, sorry.
All right, so our presentation will be on confocal microscopy and phase treatment for MRSA. I'm Ryan.
I'm Miles.
I'm Eric. So in this age of antibiotics, modern pathogens have evolved to become resistant to many of the available treatments typically implemented in healthcare. And one such culprit is methicillin-resistant Staphylococcus aureus, or MRSA. However, researchers are trying to exploit a certain cellular feature, which are membranous protein receptors. And such molecules are used to connect to other receptors or to allow cellular intake of substances. And by engineering the tail fiber proteins of phages or bacteriocins, such as the R1 pliocins, scientists can gain another tool to combat the spread of antibiotic resistance and use bacterial features against these pathogens.
Antibiotics are extremely important tools in fighting bacteria in humans and animals, and their usage has been increasing rapidly in recent years. Not only are antibiotics used in medicine, but they are also used in other industries such as food production, meat production, horticulture, and more. The overuse of antibiotics and the ability of bacteria to mutate has led to antibiotic-resistant pathogens. Some of these viruses are exceptionally difficult to treat, particularly the MRSA superbug. The global crisis of this antimicrobial resistance has reached a point where, if the problem is left ignored, the world may enter a state where simple injuries, such as infections from small wounds, could once again be life-threatening. Recently, this problem has spurred increased efforts in this fight against these deadly antibiotic-resistant pathogens. We propose a particular solution in combating the MRSA infection involving viral engineering and the use of a technique known as confocal microscopy.
Okay, so the bacteria we're studying is a methicillin-resistant Staphylococcus aureus, or MRSA for short. And this bacteria can cause life-threatening infections in the bloodstream. It's an invasive bacteria, meaning it can attack parts of the body that usually don't get bacterial infections. And like many other types of cells, MRSA has receptor proteins on its outer cell wall, known as cell wall anchored proteins, or CWA for short. So we're focusing on a type of CWA protein called NEAT, which stands for near ion transporter proteins. And these proteins have critical roles in Staphylococcus aureus, as they take in heme and iron, which are important nutrients for the bacteria. In addition, recent studies have shown that MRSA with high levels of the CDI-AMP signaling molecule is more resistant to antibiotics. CDI-AMP is critical for MRSA growth and division. So to attack bacteria, scientists have engineered a type of bacteriocin called the R-type pliocin, which can bind to specific bacteria and inject chemicals to kill them.
In addition, bacteriophages, which are common viruses that kill bacteria, have been engineered to target specific bacteria. Potential candidates for this includes members from the Cautoviralis family.
So in addition to the previous information, we can apply what we know about confocal microscopy as a way to facilitate MRSA treatment. So confocal microscopy is a subset of optical microscopy to produce high-resolution images. It achieves this by using a pinhole aperture to block out unfocused fluorescent or laser light. And the microscope then takes a series of pictures from one focal plane at a time, and this process is repeated for a varying amount of time. And the more scans per focal plane and the more repetition, the more detailed the micrograph becomes. And during this procedure, the microscope, in addition to a 3D rendering program, stacks each of the scans into a three-dimensional Z-stack. Depending on the program settings, the microscope can produce clear and detailed images of the sample. It also removes the traditional limitations of optical microscopy in a sense of being constrained to a two-dimensional plane. So researchers have used a technique to map out the topography of cells, and this existing information can be applied to research the receptor proteins on MRSA and match the corresponding bacteriophage or bacteriocin tail fiber proteins.
So in our solution, we propose that confocal microscopes can be used to map out and analyze the entire bacterial membrane of the MRSA bacteria. By mapping it, scientists can then determine the locations of neat protein receptors and CDIM binding sites. With a comprehensive model of the MRSA bacteria, scientists can engineer R-type pyocytes and bacteriophages by modifying their tail fiber receptors to correspond with bacterial membrane proteins and target specifically neat protein receptors and CDIM binding sites. These R-type pyocytes and bacteriophages can then be injected into human bodies where they can be used to combat the MRSA virus. And this bacteriophage treatment can also be combined with the usage of antibiotics. One possibility for the development of these engineered bacteriophages would be to run experiments on mice to determine efficacy and effectiveness.
So bacteriophages and bacteriocins like R-type pyocytes can be engineered to target specific bacteria by manipulating their tail fibers, which are the things that actually attach to the bacteria. MRSA's heavy reliance on both the CDI-AMP signaling molecule and neat protein channels can be used to our advantage. Using bacteriophages or R-type pyocytes, we can engineer them to bind to CDI-AMP receptor proteins and or the neat protein channels. Thus, we have two ways to combat MRSA. Bacterial diversity can be introduced into depleted microbiomes, allowing for a more varied gene pool. In addition, the chances of reinfection or infection by other resistant bacteria will decrease. As a secondary failsafe, these phage/bacteriocin treatments could be supplemented with antibiotics. At this point, antibiotic-resistant pathogens like MRSA have to choose resistance to the phages or resistance to antibiotics. Choosing one correlates to a decrease in the other, allowing researchers to take advantage of this bacterial evolution. Because of this, scientists don't necessarily have to develop new antibiotics, and they can instead use traditional ones, saving time and money.
So with all this in mind, there are some issues, questions, and developments for the future. So the first is, can confocal microscopy be used to develop monoclonal antibody therapy for antibiotic-resistant pathogens? Because the same topographic techniques used to find matching receptor proteins for bacteriophages and bacteriocins can also be applied to antibodies. The potential challenges will lie in identifying the exact antibody that can help neutralize pathogens like MRSA, but the groundwork for this research has already been established with COVID-19 monoclonal therapy. Our second question is, to what extent will bacterial evolution prompt changes in phage therapy? Because bacteria are living organisms, and they evolve and adapt to inhospitable conditions. Phages respond to this as well, changing tail receptor proteins to better reproduce. And as genetic changes accumulate over time, scientists will have to continue modifying phages and bacteriocins to continue to keep antibiotic-resistant microorganisms in check. And the final question is, does bacteriophage treatment run the possibility of transferring antibiotic-resistant genes to non-resistant bacteria? Because in certain situations, infections from pathogens like MRSA leaves the human microbiome severely weakened and depleted. If there is a non-resistant version of a pathogen like MRSA left in the microbiome with bacteriophages, there is potential for the bacteria to take up antibiotic-resistant genes via transformation. However, this is speculation, and more research and trials are needed for corroboration. And these are our references. And thank you.
Thank you.
Thank you.
Thank you for your presentation. Do you think can you elaborate a little bit more on how you will use confocal microscopy or light microscopy in general to help you with designing antibodies, monoclonal antibodies in that case?
Yeah, so we specifically want to use it to try to map out kind of the topography or like a 3D model of the membrane of MRSA. So when we get a more detailed model of that membrane, we can more accurately study the receptor proteins. And based on that, we can actually engineer the bacteriocins or the bacteriophages with the corresponding tail fibers that will be able to only bind to those receptor proteins so it can specifically target MRSA.
And then we can like apply the same methods to help find antibodies that can match those specific receptor proteins. But it's going to be a little difficult because there's thousands of antibodies within any given organism. So matching it to that specific receptor protein will be a little difficult, but just the topography part of it, we can achieve that by using confocal light microscopy.
We have time for one more question, if there is one.
So I don't really have a good background on this, but one question that I had was, why confocal microscopy versus cryo versus traditional versus, water vapor? I mean, they're all used to map, the structure of something you're looking at. Why confocal?
Because in confocal, you can sort of generate false color images that can differentiate between, different heights in the extracellular matrices or the receptor proteins. At the same time, it sort of, you can use a sample that's been prepared, and it's not as damaging as other electron microscopy techniques, and other types of optical microscopy, they don't produce as focused images because the confocal microscopy uses a pinhole aperture to focus the light in a certain focal plane to produce more high resolution images.
Thank you. Good job. All right, next team on deck.
So we chose infectious diseases, and our question was, how can scanning electron microscopy be used to develop better solutions for HIV? 3D representation of the HIV tissue specimen can be created by combining the electron microscopic surface images that are taken when the specimen is investigated by the microscope. This representation has enough solution to show the ultrastructural details of a cell. The monitoring of developing diseases and possible viruses in the diagnostic environment is where electron microscopy is very useful. Different electron microscopy methods such as immunoelectron microscopy and electron microscopy can also help provide better information for treatment and beneficial vaccine strategies. So our concept and topic is that the electron microscope scans a sample and it scans using the electron beams. Then these beams are launched by an electron gun, and then accelerates as they pass through the scanning electron microscope column. The electron beams are focused as they move through a series of lenses and apertures during this process. SEMs are widely used in current materials science for research and investigation into like nanotubes and nanofibers, high temperature superconductors, mesoporous structures, and alloy strength. SEMs help HIV studies because it helps like scientists and researchers better understand structural aspects of the infection, since there has been no official cure to HIV since it infects a human's body and integrates into the host genome of many cell types. The first figure is just a picture of HIV particles under a SEM microscope, and that it shows like it's infecting a human T cell.
Okay, so for the presence of a scanning electron microscopy, or SEM, it projects and scans a focused stream of electrons over a surface to help create an image, and then it's used to investigate the microstructure and also the chemistry of the materials. After that, a beam of electrons is produced at the top using an electron gun. Then it follows down a vertical path through the microscope that's held in a vacuum. And then next, the beam goes through the electromagnetic fields and lenses that that focuses the beam down through the sample. And finally, the electrons and X-rays get ejected from the sample, and then it gets converted to a signal and then converted to a final image. And in figure 2, you can see a drawing of the electron and the X-ray optics of an SEM and all the parts that are labeled.
To give a bit of an explanation on HIV, or human immunodeficiency virus, is an infection that's transmittable among people. And there are many different ways that it can be spread, mainly through contact with infected bodily fluids, needles, and etc. Symptoms for HIV may start within 2 to 4 weeks after the virus enters the person's body, and some may experience an illness resembling of the flu, also known as primary HIV infection. Symptoms for this include fever, headache, and muscle pain. Later on in the disease, clinical latent infection may occur, or chronic HIV. The body's immune system is slowly destroyed, and mild infections may occur. If HIV is untreated for up to 10 years, it may develop into AIDS, which is acquired immunodeficiency syndrome. AIDS can increase likelihood of developing severe illness that wouldn't occur in a healthy immune system. Symptoms of AIDS can include sweats, chills, and a recurring fever. And in figure 3, this is an image of a tissue sample containing mature HIV forms. And our proposal is to use scanning electron microscopy to discover the morphological features of the HIV virus in order to develop more advanced cures for it. In the past, scanning electron microscopy was not used very often to study microbiology due to difficulty with preparing specimens, but as higher quality tools have been developed, like polycarbonate filters, and they're available now, they are very effective in collecting viruses with pores that can scale all the way down to 10 nanometers. And these filters aid in surface observations of pathogens such as viruses and bacteria. This entire SEM process for imaging microbes requires a conductive surface for good contrast and reduction of charging for very small organic particles. Thus, the virus sample will need to be coated with a sort of ionic liquid so it can have an electrically conductive surface, and it can reduce specimen charging. The virus also requires to be frozen at a high pressure to reduce ice crystal formation and thus specimen damage. These procedures are not conventional to usual SEM imaging, but are more optimized for biological samples undergoing it. And through these procedures, we will be able to study the morphology and structure for HIV and potentially come up with effective treatments that can lessen the risk of it developing into AIDS.
So what does this mean for the future? Well, by increasing the study of HIV using the scanning electron microscopy, a few advancements can be made in the field. So first of all, more effective treatment can be made for the virus, like improved antiretroviral therapy, and a potential cure can be created with further study. And SEM can also aid in more efficient studying of the HIV virus since it is quite small, which the electron microscope can perfectly handle. But most importantly is the development of an HIV vaccine, because despite the virus being a threat to millions of people throughout the years, a vaccine still hasn't been developed. And with the creation of an effective vaccine, the rates of HIV contraction can decrease exponentially and increase people's chances at having a healthier life. These are the references that we used. And thank you. Are there any questions?
So, in using an SEM to study the morphology, what is the resolution if you're using an environmental SEM? And does it actually sufficiently capture the level of morphology that you're looking for?
It does capture like the sufficient morphology we need. Because like the resolution for the SEM can go very small, and the HIV virus is about 100 nanometers in size, so it would work for that.
What is the resolution for an environmental SEM? You can get back to me if you're looking this up. It's okay to not know off the top of your head. Somebody can ask another question in the meantime.
So what exactly will you be looking for in the cure with SEM? Like, do you have an idea of what you want to look for?
Yeah, the SEM will basically look for like the virus so that it can see like a closer structure of it, and then it will be able to look at the sample to get a better structure so that they can better research it to help develop like a vaccine.
Aren't there like super detailed 3D models showing like the atomic structure and everything?
Yeah, but the SEM can help to like look at it closer, because compared to other microscopes, it can see more details in the virus compared to like a light microscope, and so it can give you a better view of the HIV.
I wanted to ask you, from the literature you have been reading on HIV, have you seen any explanation why the HIV virus eluded any vaccination? Why is it so hard to come up with a vaccine?
It was difficult to make a vaccine because of the way the virus works, because it invades and it commands like the T cells that are responsible for defeating it, and so the number of functional T cells is reduced, and it also ruptures like their cell membrane in the process. And also HIV, it mutates at a very fast rate, so it makes it hard to treat with just like a single drug. And also it can hide and like latent in certain tissues and other places, so it can like avoid being detected.
All right, I think for the sake of time, we're going to move to the next group. Thank you. Oops. All right, next team on deck.
Okay, so hi everyone. The topic that we chose to do our project on is infectious disease, and we mainly focused our topic on COVID-19, and it is by Jillian,
Melody,
Leia,
and I'm Cindy.
Okay, so to start off, we have the abstract. Okay, so to start off, we have the abstract. So for our project, we focused on infectious disease, specifically COVID-19 because of its enormous global impact. Since it was first discovered, scientists have researched and gained an understanding of many components of this virus. However, there are still quite a few things we are unsure about. In order to find things that we could improve upon, our team researched unexplainable aspect of COVID-19 that so far scientists can only hypothesize about. Focusing on the phenomena of long COVID-19, our solution provides an experiment to help patient and their doctor determine whether previous disease will impact the functionality of the body.
We chose to explore how microscopy can further contribute to fighting the virus that has taken over the world, COVID-19. In the beginning of the pandemic, scientists from all different countries and fields conducted research as fast as they could to gain an understanding of what exactly this virus could do. After learning more about microscopy techniques this week, we were able to appreciate this field's hand in developing vaccines and how many lives were saved through the use of these tools. However, many questions still remain unanswered. The question we decided to focus on is how can we use microscopy to find a way to tell who will develop long COVID? Wait, I think that was the wrong slide. Yeah, it's like this one. Okay, currently not much is known about, wait, can you go back? Yeah, wait, no, back, yeah, that one, okay. Currently not much is known about long COVID-19. It has been observed in patients of all ages from all mild symptoms to people who are hospitalized. It seems like whether or not someone will have lasting symptoms is sort of random almost. Many have hypothesized that individuals with various past viruses and diseases may be related to having long haul COVID.
Before our proposed solution came along, the current state of science was already fairly advanced. While researching for viruses, scientists tend to use electron and atomic force microscopy. Electron microscopy is commonly used to study small biological structures and can be divided into scanning and transmission electron microscopy. Scanning electron microscopy works by scanning a sample with electron beams. The electron gun fires the beams, which pass through a series of lenses that help the sample go into focus.
The beams then scan the sample in a raster pattern and interact with the atoms on the surface of the sample. In SARS-CoV-2 research, this method was used to study the infectious cycle of the virus in Vero E6 cells. In SARS-CoV-2 research, this method was also more rapid and efficient than transmission electron microscopy. Transmission electron microscopy works by shooting electrons out of an electron source and then letting the electrons travel through the vacuum column of the microscope. The electromagnetic lenses focuses the electrons in a thin beam, and then the electrons pass through the specimen. The detector/camera detects the electrons, and then it takes many 2D pictures to build a 3D model. In SARS-CoV-2 research, this method controlled some key findings. Atomic force microscopy works by letting a probe mechanically scan the surface of the sample. The nanoscale tip is attached to a small cantilever, which forms a spring. When the tip contacts the surface, the cantilever bends, and then the bending eventually reaches the photodiode, which makes a feedback loop to control the constant interaction between the tip and the surface. In SARS-CoV-2 research, atomic force microscopy reveals structurally intact infectious and inactive SARS-CoV-2 upon low concentration of formaldehyde treatment. This accelerated researches against the virus. And the picture that we have up there is a picture of an electron microscope.
Our solution. So what are we proposing? We're proposing an advanced microscopy technique that could be used to predict whether someone would have lasting COVID symptoms depending on the types of diseases that they had before.
Okay, so our experiment is basically using zebrafish, and using zebrafish has a lot of advantages like very quick breeding and easy manipulation. During the zebrafish's fertilization, their one-cell stage fertilized eggs can be easily injected with DNA or RNA. During this time, we will split the zebrafish into two groups, the experiment and the control group. During this stage, we will first modify the experiment group's zebrafish's DNA so that their genes will replicate a humans with certain diseases. And then we will inject them with the virus. In the control group, we'll just be injecting the fish with the virus. Then we will grow out the zebrafish from their birth to their death. And during this time, we will study these fish to see the long haul effects and whether there is a correlation between the diseases that they had in the beginning and the virus. And we will be using optical and electron microscopy techniques to study these fish. Optical microscopy techniques can allow us to dye the different parts of the cell and also dye the virus to gain a better understanding of what is happening. Electron microscopy techniques can allow us to look at the suspended DNA molecules and also gain a better understanding of what is happening. And the picture over there is just a picture of a zebrafish cell.
Future directions. If our experiment can be successfully performed in the future, there will be more accurate predictions on the long-haul COVID-19 effect, based on many kinds of past diseases that individuals have experienced. Additionally, if scientists could find out who would be a long hauler, it could also lead information about why someone might be a long hauler. Here are our resources, and thank you for listening.
Are there any questions?
Nice talk. Thanks, girls. So, quick question. You know, we use mice a lot as a model organism because as much as they're different than humans, we can put, you know, sort of disease models into mice. Using zebrafish, I understand why, because you guys saw them in class, right? They're optically transparent. You can see through them. They're really great for optical microscopy, looking at things that are dynamic. Did you find in your research at all, you said that you're going to need to modify the DNA of the zebrafish to basically like make them appear as though they had previous human diseases. I'm not aware of it. Is that something that people are doing, right? Because we have disease models in mice and rats and other sort of higher animals, but have people tried that sort of thing with zebrafish? And if they have they been successful?
When I was researching, I have come across some articles that have said like they've modified the genes in zebrafish, and they've also said that it's actually pretty common to try and cure human diseases by modifying the genes in zebrafish so that it is like as if they had the human disease and then tried to cure the zebrafish, and it has worked in the past.
So we learned that not all animals are susceptible to COVID-19 virus, right? So I really don't know if the fish will be susceptible to the virus. Let's say it is not. Following up on what you said about modifying the genome of the fish, what could you think of doing to make the fish susceptible to the virus so it can be used as a model?
If the fish aren't, if they don't respond to the virus, then either we would use a different animal or we would probably modify like the cells of the fish.
And how could you think of doing it, for example? What is a key factor that allows the virus to go into the cells it wants to target? Have you come across this in your reading? There's a specific protein, right, that is at the surface. So maybe you can think of the same way we learned in class how you can put like a fluorescent molecule in, right, that is coded by a piece of DNA. Maybe you can think of taking the gene that will code for that specific receptor and put it into the fish, right? Yeah, okay.
Last question there.
So you said that you're going to be looking into like the long term and who's going to have like the vaccine, or no, sorry, the disease like after, like a lasting disease. But like the immune system response for zebrafish and humans are quite different. So like how are you going to manipulate that? Like, using the data you got from the zebrafish, assuming that like they are suspects, they can get the virus, and how would you transfer that to like humans and like human research?
For the long haul effects, we'll be mainly just looking at the cells and like how the cells are reacting to the virus.
Okay, well, thank you, and we can have the next team come up now.
Hi everyone. So we're group 7, and we're going to be talking about microscopy's influence on cultured meat. I'm Marisol.
I'm Maddie.
And I'm Dahlia. So livestock agriculture contributes to 11% of global greenhouse emissions worldwide, uses 30% of arable land, and consumes 50% of all antibiotics. During an ongoing climate crisis, cutting down emissions through cultured meat has been a popular option. Cultured meat is the cultivation of cell culture to make a product similar to conventionally farmed meat. It is not yet at a commercial scale due to the expense and inefficiency of current microscopy techniques, so we hope to create a new instrument that is able to view the cell culture as it grows. With our new technology, scientists could use the information collected to scale up the process.
So this is our topic. So cultured or lab-grown meat is meat that is produced by in vitro cell cultures of animal cells. And the goal of this is to limit the problems involved with industrial livestock farming. And to make cultured meat, scientists replicate conventionally produced meat through stem cell and tissue culture. So here's like a diagram. So first you get a cow and you take some of the cells out of it. And then these companies could sell it to like some sort of, sell to a medical lab or a lab of some sort. And then they're able to grow the cells, which produce muscle tissue, which is like basically the meat you eat in like a burger. And then, yeah, you can eat it. And then this is Jason Matheny, and he founded New Harvest in 2004, which is a research institute that focuses on cultured meat and animal product alternatives. It's a nonprofit organization promoting research into cruelty-free in vitro meat and other meat substitutes.
According to Oxford University, lab-grown meat could cut down the amount of greenhouse gas emissions of livestock agriculture by 96%. Emissions of greenhouse gases such as methane, nitrous oxide, and carbon dioxide contribute to significant global warming. Emissions also cause ocean acidification, since our oceans soak up to 30% of CO2 in the atmosphere. And acidification of our oceans is an increasingly more concerning problem because it causes animal shells and skeletons made of calcium carbonate to dissolve. Furthermore, according to the University of Colorado Boulder, switching to lab-grown meat can cut down water composition by 82 to 96%. Global animal production requires 2,422 gigameters cubed of water every year, 90% of this water being feed for livestock. Much of that water is saved by not farming meat the conventional way.
So this is the greenhouse effect. So the greenhouse effect is when energy from the sun warms the earth, and then some of the heat is trapped in the earth and is held by the greenhouse gases. And this effect is needed because the earth normally is 60 degrees Fahrenheit, but without this atmosphere, it would be 0 degrees Fahrenheit. But due to greenhouse gases such as methane that cows produce a lot of, they trap excess heat. Due to the atmosphere layer like getting bigger, they trap more excess heat, which heats up the earth too. It's heating the earth too high.
Okay, so although cultured meat is hopefully going to be more sustainable in the future, as of right now, here are the cons of cultured meat, which we hope to improve. So first off, cultured meat is not yet sustainably scalable. So basically, like in the stage that it is now, it is not at the point where we can completely commercialize it and like send it off into the world so people can buy it. It is also difficult to make, especially when it comes to larger pieces of meat. And it is also unknown how the general public will react to technically lab-grown meat. Companies within the meat industry could falsely advertise this lab-grown meat, which could cause stigma and convince consumers that there is something wrong with cultured meat, when it basically is pretty much the same thing as the meat that you'd get from like a normal animal. Okay, so currently there are many studies and tests being done looking for different ways to create scaffolds for cultured meat to grow on in the hopes of finding something sustainable and scalable, since scaffolding is essential to the growth and development of cultured meat. Essentially, the cells must be transferred to a scaffold to produce structure, and which can help like make thicker meat products. Depending on the material you use for scaffolding, it can have an effect on the structure and even the taste of cultured meat. Some examples of scaffolding biomaterials include gelatin and soy milk, alginate, cellulose, and spider silk.
So how is microscopy used to observe behavior of cultured meat? Scientists have searched for a specific protein while immunostaining the myosin protein to make sure the cultured meat would be similar to farm meat. And for clarification, immunostaining is when you use antibodies to detect certain proteins in a substance. Then they looked at this myosin with a confocal microscope. Current cultured meat research involves spectroscopy to measure glucose and metabolites. And you see on the side, that's a graph from an infrared spectra of water, carbon dioxide, and ethanol. Although that's not one that would be used for meat, that's basically an example of what it would look like to identify molecules. And that's necessary because to recreate actual meat from animals, there's many molecules that go into it and not just muscle tissue. So with spectroscopy, they measure glucose and metabolites. And there are methods of measuring glucose and metabolites using enzymes, but they're not very efficient due to the fact that they use the glucose in the sample. Scientists use the spectroscopy to identify molecules using fluorescence, infrared, and Raman spectroscopy. And they use this system because there's no direct contact between the equipment and the sample, which is ideal for soft tissue cells. The vibrations from infrared and scattering of vibration from Raman or photons from fluorescence are better ways of analyzing the tissue, and unlike the enzymatic methods, they do not eat away at the sample. Using spectroscopy to identify molecules of interest can be difficult because of the wide range of molecules, and the fact that the molecules have low levels of concentration. It also becomes inconvenient because live tissue cells are always changing and growing. Spectroscopy can be utilized to find the density of the material in addition to bacteria present. So spectroscopy shows these variables by scattering light through the sample. However, none of these methods are reliable when it comes to determining how the molecules would perform commercially and can be affected by outside variables like air bubbles or foreign particles in the cell culture.
Okay, although the previous methods of microscopy were very useful in terms of making groundbreaking discoveries when it comes to cultured meat, these techniques are not perfect.
So with confocal microscopy, scientists are able to make 3D images of molecules and cell cultures. And if we were able to upgrade or just use this technology with growing cultured meat, we could look at each stage of cultured meat and see the difference between it and how normal meat grows, and then compare them in order to make it on a scalable level that we could all eat.
Okay, due to cultured meat being new to research, we wanted to focus more on the research aspect of cultured meat and less on creating a final product of industrialized cultured meat. Our idea is to have a bioreactor to observe cultured meat as it is growing in larger amounts. The way that this might work is that there would be microscopes embedded into the bioreactor that can take samples of different areas of growth, so we can see the meat growing in different areas around the same time. This method would be a much simpler way of experimenting on different biomaterials used for scaffolding to find the one with the least flaws. This bioreactor can also be applied in other areas of study, not just cultured meat and maybe even cancer. And those are our references. Thank you.
So apologies for missing some of the beginning of your talk, but I'm actually curious if you thought about using scanning probe microscopy for your bioreactor as well, because I would think that maybe there were some interesting opportunities to see textural changes with that technique over time that might be useful.
We thought about that, but it's not the best option because these cells are so delicate and fragile that if you were to use something like a really sharp needle, it wouldn't go without damaging the tissue, and it just wouldn't be a very good way of getting an image since it would just be damaged.
Do you think that maybe one of the non-contact modes might work? And if not, maybe think about, or do you have an idea as to why?
Yes, that's why we decided on confocal microscopy because it creates a 3D image. We also talked about how all the other methods being used right now require no contact, and that's ideal, which is why in the end we chose no contact.
In your cons, you mentioned how like you don't know how consumers are going to react to like cultured meat. So is there like a way you would sort of market it and just so it's like more appealing to like the general public?
Well, I think like with like GMOs and stuff like that, we can just try to educate people and show like the science research and just hope kind of for the best and hope that people understand that this is like would have such a positive impact on the environment as well as like it's still the exact same thing.
There was also, you might remember from one of the articles we read, although this is a little misleading, when presented with less cons, people were more likely to see it in a positive light, which, you know, that's what you gotta do. If you look at meat today, it's not like they mention any of the cons that go into it, even though there's so many more than ours have. So it's not And when you think about it, like saving the environment is really important to people, especially when we're seeing the effects right in front of us. So I think the urgency of our environment crisis will make people think more critically and see past that.
Great. And then one more question.
So I'm not super familiar with the processes of growing cultured meat, but you mentioned that one of the challenges was sort of getting like a, I guess like a large chunk of meat to grow as opposed to just small pieces. So I'm wondering if that's related to, I'm thinking maybe like metabolism challenges, like getting nutrients of, like the bigger you go, these cells on the inside, they have less access to that. So could you guys maybe comment a bit on that and perhaps how that could be resolved?
In terms of getting large pieces of meat, we didn't see problems as much with nutrients. I mean, with our research, it talked more about scaffolding and how without a proper scaffold, which is still, they still need research for that to figure out which one will actually work the best with these cell culture. So it's more of a matter of what we can get these tissue and like cell culture to stick to and to want to grow on, I think.
Yeah. All right, thank you. All right, teammate.
Thank you. Hello, we'll be talking about the applications of TEM in the field of electric vehicles. I am Ethan.
I'm Louis.
I'm Kyle.
Just to begin, there are a lot of concerns about electric vehicles that I want to go over. First of all is mileage. EVs get relatively low mileage compared to the gas sedans of normal, are relatively like equal size cars. If, for example, the Tesla Model 3 right now gets around 300 miles on a full charge, and then a gas car would get around like 480. Safety, lithium ions are known to explode. You know, you see some of those videos online like, oh, Tesla explodes, that happens. Charging takes a long time, and also you won't find it, you know, around the country a lot. You know, we live in excuse me, California, which is a lot of chargers everywhere, but like, you know, if you go to somewhere in Texas, I don't think that there will be that many. And then price, EVs are expensive. The Model 3 right now is about 40 or 45,000, and then gas cars are around 25,000. And the repairs are also pretty bad. You know, some people might be sad about the price, and some people might even just exploit it for content on YouTube because they'll make them more money than repairing the car.
So concept and topic, state-of-the-art characterization techniques and its applications help us understand the high-performance LIB, which are just the standard lithium-ion batteries and beyond lithium-ion batteries. These characterization techniques have been based around in situ and operando designs, which have shown their usefulness in battery research. In addition, electron electron microscopy techniques such as TEM, which is transmission electron microscopy, have also provided temporal and spatial resolution on nanoscale electrodes. TEM has also allowed for real-time information and experiments on electrode applications. And TEM is also used to study in situ and operando cells for electron configuration. And in situ and operando characterization techniques have led to a decrease in corruption and permanent damage in technology.
So this is just a general rundown on how batteries work. So for the model cell, or the example, I'm just using gold and lithium because lithium, obviously that's like the primary battery because of the high voltage and the oxidation reaction. And gold, I just included that because it was easy. So basically, the electrons just are generated in this formatting. Got a little wonky here, but
the electrons are generated in this half reaction and are separated to form ions, and electrons pass through to the other one, and that flow of electrons is what powers the cell. And I mean, there's more specifics like the salt bridge, like the two ions, which are aqueous, go to fill the either cation deficiency or cation F or SX. But yeah, this is just basically a rundown of how the cell works. And then, so yeah, batteries like this use that, and it's depleted once this kind of runs out, either one of the reactants, which is gold or lithium solid, gold ions, lithium solids. So yeah, that's basically a rundown of how it works. All right, so the microscopy process for this is, so the microscope just shoots light through the sample. Let's see. And in electric vehicles, this allows like the imaging of the actual power cells, and this can help identify the batteries like structural flaws, which can damage the battery, cause like mistakes in it, or limit its battery life. And if we can see these mistakes and fix them, then we can better optimize batteries.
So how microscopy helps us, Kyle touched on this a bit, but I'll go a bit more in depth. Basically, chemists and engineers can view the contents of the batteries on an atomic level. So this part, I believe, is the cathode, and this is the anode. And this little buildup are the anions and the cations, respectively, building up on the things. And research can reduce the explosions, because what happens is, as I'll explain in this next slide, what happens is that when the anions go onto the anode, or in this case the lithium anode, they will not form correctly, and it will form these little dendrites. Not to be confirmed with the biology dendrites, these are just called dendrites because of how they look. And then they'll actually interact with the separator, which is also conductive. So any bit of electricity, if it hits the separator, will cause the dendrites to pretty much explode. So then use of graphite can reduce dendrites, but it also decreases lithium available. And the graphite also impedes the charging, and faster charging also leads to more creation of dendrites.
So recent advancements in batteries. So a recent advancement has been the development of nanobolt lithium tungsten batteries, which have been strengthened with tungsten and carbon nanotubes to the anode section. So it provides more energy. And the addition of this lattice, which is a regular repeated three-dimensional arrangement of atoms, ions, or molecules, increases battery efficiency, as the nanomatrix that is added to the anode creates a larger area for the electrons, which results in shorter charging times and longer battery life. So if we go back to the hurdles, the issue with the disorderly growth on electrodes and dendrites is solved with the addition of tungsten and carbon nanotubes. The issue with the issue with energy efficiency is solved with tungsten and carbon nanotubes as well to the anode. And the issue with lithium use being decreased is also solved with tungsten and carbon. And the increase in area for the electrons provides faster charging, but removes the issue of dendrites, as the smoother surface removes the disorderly structure of dendrites. Here's our works cited.
Yeah, so that's basically it. Is there any questions?
Environmentally, how would you address the problem of e-waste? Because I know lithium-ion batteries can be like an alternative for other like natural gases, but how would you also address the e-waste problem and the toxic metals within lithium-ion batteries?
I'm gonna be real honest with you, I didn't research that. There are other ways, like putting them in, like this is not like the way that I would endorse, but if you put it underground, it will eventually decompose, or other stuff would happen. But there are ways that being researched right now that I'm pretty sure will solve this problem.
Can you go a little bit more in depth about the costs of using the different materials? Like, what is the most cost-effective?
Yeah, so can you go back to the slide? Yeah, right there. So tungsten and carbon nanotubes, although like you're adding more materials, it's actually more cost effective because you're decreasing the use of lithium batteries.
So these dendrites that you're talking about, did you guys, in your research, did you guys come across any method perhaps where you can eliminate or maybe stall the growth of these dendrites through some like non-invasive procedure that you can treat the battery with? Just wondering.
Oh, do you mean like repair dendrites?
Yeah, or like reverse the process, or perhaps somehow removing them maybe through, off the top of my head, I'm thinking like dedicated sonication, like focused sonication or something like that.
Yeah, we didn't research that particularly, but I believe that if we eliminate the cause in the first place and stop these things from actually growing by having a smooth layer on the anode to build like the anions, then I feel like we won't have the problem with dendrites and won't have to actually go into the battery to destroy these. Thank you.
Yeah, so our last team, one of the members is on Zoom, so this might take us a little bit of an extra minute, so please bear with us. So let me see. So, hi Asher. Can you talk again so we can check the volume?
Hello.
Okay, good, we can hear you. So do you guys maybe want Asher to present your slides and share his screen? That might be the best way of doing it. But what do you guys, did you guys already talk over this?
We can switch the slides for him. Yeah.
Okay. Okay, that sounds good.
Although, Asher, you'll have to tell us when. If that's all right with you.
That's fine, but I can't see I can't actually see the screen, so I wouldn't know like what slide you're on.
We should be able to share the screen. So I'm gonna just do this like that. So I'll put you here, I guess, so we could see you, and then put your slides. Over here, and then that's not quite.
Can he see the slides?
Yeah.
We can all say hello. We don't need to see it necessarily.
Okay. Well, I'm putting you up for a second so people can see you for a second, and then I guess we're just gonna have you here, and then I'll do share screen once the slides are up. So let me just get these going. He can't yet because I haven't shared my screen, so I'm about to do that. Okay, Asher, can you see the slides? Okay, perfect. Then I will turn it to you.
Is this on? Okay, good. Hello. Our presentation is on infectious diseases. I am Oliver Liu. This was done in collaboration with Andrew Herr and Asher Marone, who is unfortunately joining us on Zoom today. Let's see.
Try the slide advancer. That might work better.
Where is that?
Oh, maybe.
Okay.
Yeah, you guys can probably advance. I mean, it's not the most efficient, but you could do that.
Okay, all right. Yeah. Right, so the rate of antibiotic resistance is increasing all throughout the world, right? This is a major problem due to the fact that antibiotic resistance, when bacteria have it, it means that antibiotics simply do not work. And that leads to increased mortality rates and a decrease in quality of life of the people who are affected. Now, antibiotic resistance isn't the only problem with our current usage of antibiotics. Now, what our antibiotics do is that they don't only target the bacteria that we want to get rid of, right? They target the entire human microbiome, right? And a lot of these bacteria are beneficial to our body, right? For instance, they might catabolize food for us or produce beneficial amino acids, right? So these problems with the antibiotic treatments that we currently have makes developing a more specific treatment for bacterial infections all the more important. And so that's where piacins come in, right? Piacins are a bacteriocin, or a specific antimicrobial peptide produced by bacteria in the Pseudomonas genus. They're generally used in nature to eliminate the Pseudomonas' competitors so it can prosper. There are three classes of piacins that we currently know about, being the S-type, the R-type, and the F-type piacins. And they're all currently being researched for their potential uses as targeted antibacterial agents. And okay, right. And now I'll introduce some of the concepts. Right. So the creation of targeted antibiotics can be assisted by the use of electron microscopy. So for example, the protein structure of piacins can be investigated and recreated. And understanding the biochemical structure can, of course, provide insight into how to apply different engineering techniques to these peptides. And also, obviously, improvements in antibiotics will benefit the field of medical science. Right. No, that's not it. There we go. Go ahead.
Okay, so currently, modern antibiotics, as previously mentioned, they kill both good and harmful bacteria. And normally, this isn't really a major issue, as normally it involves the fast and speedy recovery of the patient. However, in some extreme cases, using an antibiotic can actually allow a disease to infect the host. So to further illustrate this, Clostridium difficile is an infectious disease found in the intestinal tract. And normally this disease will want to find a place to reside alongside the walls of the intestinal tract. However, good bacteria obstructs the disease by occupying this space entirely. However, patients who use an antibiotic will kill this good bacteria and thereby free up some space. And this will allow some resistant strains of Clostridium difficile to infect the host, as seen in the picture. Okay, so to prevent these scenarios and situations from occurring, my team and I have decided to turn to targeted antibiotics, or specifically engineered piocins, which is actually a very simple process. So first, scientists will retarget the spectrum of R2 piocins by swapping their tail fibers. So halfway along the tail fiber is something called the N-terminal. And basically, scientists will fuse the N-terminal of the R2 piocins with the C-terminal of P2 tail fibers to bioengineer what's called AVR2P2 piocin. And basically, this is a beneficial and beneficial piocin that will target harmful strains of E. coli. And this is just one of many examples of how engineered piocins can be used for targeted antibiotics.
Asher,
so just before we move to the solution, just to summarize the problem, antibiotics are damaging and can lead to the Clostridium syndrome that we mentioned previously, and then also antibiotics also target helpful parts of the microbiome. Next slide. So, oh, sorry, I can't see the text.
Sure. Is that right? Which one?
There's the text for the solution slide isn't showing up.
Oh, okay, sorry. Is that it? Oh, okay.
So our solution to the issue that antibiotics aren't targeted enough would be to reprogram other phages. So the study by Dr. Scholl that my group mate previously mentioned is an example of how bacteriophages from the Pseudomonas aeruginosa could be used to target E. coli. So our idea is basically just to use that same method of adjusting tail fibers to land on the membranes of specific pathogens, and just apply that to other other bad pathogens, to other infectious diseases, and use that to target the diseases instead of general antibiotics that could cause issues to the microbiome and could cause Clostridium. Next slide. And so just like on the future directions of what we expect here, we expect that there's still a lot of questions to answer with regards to the feasibility of modifying bacteriophages to target every disease. And we know that there are limitations to the solution because testing modifying the bacteriophages for every disease would probably take a very long time. So it might not work for every disease immediately, and it could take, up to several decades to really develop these bacteriophages for every disease. But it's definitely a possibility, and something that really should be experimented on, because it does really enable a good solution for more targeted antibiotics. And to relate this to microscopy, a major barrier to Dr. Scholl's experiment was that they couldn't actually look at the S-type piacins because they were too small to see with microscopes. So hopefully and they used an electron microscope, and the piacin was too small to see. So hopefully over time with advancements in microscopy, it would be easier to re-engineer these phages to target specific diseases. And I think, next slide.
Yeah, these are references, and thank you.
Are there any questions?
So I know everyone wants to go eat. It's always, being the last group before lunch, right? So you guys heard a talk, obviously on this topic from Professor Miller. In terms of you're going to re-engineer the tail fibers, and you want to use EM, right? What you saw during his presentation was using EM to understand the actual mechanism of how these things operate. What would you use EM for in this case,
right? We would be using EM for determining the structure of the tail fibers of the piacin. Yeah. Oh, there.
Any other questions?
Who had a question?
So I know with the EM microscope you can see the tail fibers. How would you actually change them so that it does go for specific bacteria?
Wait, sorry, can you repeat the question?
What's the actual method for changing the tail fibers so it looks for specific bacteria? Because I don't think EM can actually manipulate it, can it?
Yeah, no. So we're actually using the electron microscopes because obviously, as you mentioned, it doesn't specifically reprogram the tail fibers, but we're actually using it to examine the structures and identify just how it works in processes.
So you might be able to modify the tail fibers by using certain chemicals, right? Because what you're trying to do is cleave part of a tail fiber off and attach a different portion on, so you can do that chemically.
Okay. Oops, I need to use the mic.
There's a mic.
Here's a mic. All right, so thanks again to all the students. Let's give all the students a collective hand on their presentations. I would also like to thank a couple other folks. So you saw talks on 4 different topics, and those topics were provided to the students before they got here as 4 sort of key areas of science and technology. And we heard on Monday very sort of dense lectures from some faculty members here. And so I'd like to start out by thanking, first of all, on the topic that you just heard about, which is infectious disease, CNSI Director and Professor Jeff Miller for taking his time to give us a talk. We also heard in the topic of batteries, which I think was before that talk, Professor Yuzhong Li from the Chemical Engineering Department came and gave a talk on how he uses EM for battery research. We also heard from Professor Katsushi Arasaka from the Department of Physics and Astronomy about the BRAIN Initiative and his models and ideas about how to use optical microscopy to better understand the brain and consciousness. And then we heard from Kathleen Chen, who's a senior graduate student in the ROAT lab, which is Professor Amy Roat is a professor in molecular, cellular, and developmental biology, studying cultured meat, right? So those 4 topics were provided to the students ahead of time. They had literature that they read about. These are sort of grand challenges areas in science that map onto the use of microscopy, so that's where the thematic things come from. So I'd like to thank all of my colleagues for taking their time out to come and speak to the students. In addition, Rita mentioned that there's, it sort of takes a village, right, to run this program. There's some other folks that helped us out throughout the week, mainly a number of graduate students who've helped out with all of the hands-on demonstrations. So Jason is here. Jason Hu is here. Also helping out, and he's been here quite often throughout the entire week. Emma Valinsky has also been here helping out throughout the entire week. And then some of the other graduate students that have helped out, Bruce Liu, Jonathan G, and Yuyun Chen. And then there are, in addition to the professional staff that you saw on the slides, we also had 3 of our other professional staff here helping out through the week. So Michael Lake, Matt Schibler, and Paul Chang have also been, people on the ground, excuse me, working with and showing your students, all the different details and hands-on activities. So it does take a whole group of folks, as well as the people in the booth, Mark and Mitch, and all the events team and everybody else who makes this sort of thing happen. It wouldn't happen without them. And so we really hope that you had a great time here for the week. It's a unique exposure to a place like UCLA. As I said yesterday at the end of my lecture, hopefully we'll see some of you here in a year or so. You know where to find us. So if you do end up back at UCLA, please come by and say hi. But in terms of saying goodbye, we're going to head upstairs and have some lunch on the 6th floor patio. All the students know where the 5th floor is. It's 1 floor above where we were before. But before you head out, there are also T-shirts for you outside here when you walk out the front, right? And the T-shirts are actually pretty cool, right? We have some nice design aesthetic from some of our team. Rita is a fantastic designer as well as being a great scientist. And so please grab a T-shirt. And the last thing I'll say, I just saw a whole pile of emails come through from Paul. Those of you who sacrificed 1 strand of hair yesterday for the AFM measurement, he just sent you all the images of your hair. So, you have another souvenir to take with you. So with that, we will all stop talking. Thanks again for a great week. Best of luck in whatever the next steps are in your education and career. We hope to see you all sometime soon doing great things. So please take care.